Should I get started, Jessica?
Yes, you can go ahead and get started. Thank you so much.
All right. Hello, I'm Hugo Bellin. I'm the president of the Genetic Society of America, and I'm really excited to welcome you all here today for this GSA Awards seminar. Today's awardee is Edward Smith, who's receiving the 2021 Elizabeth Jones Award for Excellence in Education. Ed is recognized for his outstanding contribution to research training and genomic education. Alongside his distinguished research program in poultry genetics and genomics, he has dedicated much of his career to broadening participation in biomedical research and helping minoritized students thrive in scientific careers. As well as training undergraduate and graduate researchers in his lab, Dr. Smith has led 2 NIH-funded research education training programs at Virginia Tech designed to increase the numbers of students from underrepresented groups pursuing biomedical PhDs. To date, these efforts have resulted in 81 PhD graduates, of which 11 are now in tenure track or tenured positions. He also led an experimental learning programs for K-12 students and teachers and worked tirelessly to prompt discussions and actions on issue of diversity, inclusion, and bioethics in genetics. Ed received his bachelor's degree from the University of Sierra Leone, received his PhD in Tom Savage Labs at Oregon State University, and did his postdoc with Susan Lamont at Iowa State University. Before joining Virginia Tech, he was an associate professor at Tuskegee University, where he was instrumental in bringing a comparative animal genomics program to the university. I have quotes here from Margaret Warner Washburn, who nominated Ed for this award. Ed Smith is single-handedly transforming science by opening the door to so many students who now feel they are part of the scientific enterprise. And there is a perspective directly from one of those students, Anjoli Diaz, who was one of the first to enter these training programs at Virginia Tech. And he's now an associate professor at Ball State University. Of Ed, he says he would advocate for each and every one of us. He was someone that would always be able to we would always be able to turn to if we were experiencing barriers that we didn't know how to maneuver. And of the program Ed helped create, she says, he was one of the best things that has ever happened to me. Even though we can't hear your applause out here, I know you will all join me in saying thanks to Ed Smith for his service. I'm not going to turn it over to Ed, but before I do that, I would like to say that we're all under a code of conduct, and Christy will share that code of conduct in the link. And if you have any questions, please go to the chat box and post your questions there, and at the end of the talk, I'll confer these questions to Ed. Ed, take it away.
Thanks, Hugo. I don't recognize myself in that introduction, but I've been in this business for a long time. So thanks to the society also for this award. It's really incredibly humbling. You know, from where I came from, this is not something you dream about, right? So I've been telling my friends that this is a man bites dog story. So thank you. And thanks to everyone, Christy and Jessica, for organizing this. This is really an honor, and I'm excited to tell you about what we've done and what maybe led people to award us this, prestigious award. So the award is named for Dr. Elizabeth Jones, who was a professor at Carnegie Mellon. And I think her distinguished career is really more impactful because she also had a lot of service that she did to the society and she served as the chief editor of our journal for several years. So I'm really humbled and really proud to be receiving this award today. I think her stamina and grit are important to not just us, but to younger people aspiring to become scientists and genome scientists especially. So what I will do today is try to provide my talk in 3 sections. So I have titled this talk, Culturally Aware Research Education. Pay attention to the differences. So we are geneticists, we have to pay attention to those differences, but culturally aware research education, it's sort of a play on what is out there that sociologists have told us to do, culturally aware mentoring. I think research education is important because part of the things that we are committed to as scientists is to train the next generation. So for this talk, I'm going to try to divide it 10 minutes each. First about culturally aware research education, and then tell you about why differences should be paid attention to by geneticists. And then this is the third part of the of this seminar, this award talk is really more recent. I've been thinking a lot about this award and this talk. A friend of mine, I stole this from a colleague of mine at Tech. We just completed interviews for our 19th cohort for the PrEP program. You will learn about the PrEP later. He will ask the candidates, you know, when you get your PhD and you have that superpower, what will you do with it? So I decided to make the third part of this talk about superheroes, what scientists are. I mean, right now we're all feeling better. I've gotten my vaccination because scientists stepped up, you know, and created the vaccine in a really very short time. But for scientists, the normal heroic charges are different. I mean, you have the superpower to recruit students, you have the superpower to train them, you have the superpower to send them to amazing careers. So the third part of my talk will focus on that power that we have and really to advocate, at least to argue in this talk, that the concept of care or culturally aware research education should be undertaken by scientists. So we should be the leaders of this effort. I am not going to provide you detailed statistics about the gaps in access to biomedical education or agricultural education. 2 days ago, the NSF released a really wonderful report that they normally do every 2 years. You can find that there. I mean, the academy still has only 9% of the faculty that are minorities, you know, and the population is 33%. So the need is there. I'm not going to focus on that too much. And in the past, I have argued about things, and this is a corny title, several years ago, 20 years ago now, actually. I argued that genomics has to be made accessible to smaller schools. I was biased at the time. I was at Tuskegee, and I, you know, made a case. And you'll see later that my my argument was taken seriously, especially by my superhero at the Human Genome Institute, Betty Graham. Okay, so the dictionary definition of cultural is when you create ideas, customs, and social behavior that are related to a society. I mean, the culture of science is you have a lab, you have a department, and you have a society like Genetic Society of America that I belong to. I also belong to AAAS. I belong to poultry science. And things happen there, right? You have to have members of the society. They speak a different language. The Genetic Society speaks about genetics and differences. So what I have done through my career is to try to say, as a scientist, the first culture that I belong to is the culture of my lab. Are we producing data? And is that data valuable in that? Can we publish it? And then the next question you normally ask is, who is doing the research? So now you have a question that says to you, okay, you're doing what scientists do. You're creating data. You are interacting with people. You're recommending people to become scientists, and you are judging grants. So that's the culture of science and the field that some of us have pursued, the profession that we're pursuing. So for us in this training grants, what we decided to do was, okay, we're going to create a culture where we can celebrate each other. And during those celebrations, we can ask them to practice talking about their research. We could ask them to practice their defense, dissertation defense. We could, give them an opportunity to practice interviewing for our jobs. So, and we do that a lot around food. And this is a recurring theme for me. So the photo on the right is a cohort from back in 2004. And the cohort here is from 2017, 2018. So it's a mix of different disciplines and of course from different backgrounds. So that's the culture that we have. And one of the values of this culture that we've created for this community is that they function without us. Even when we don't provide pizza, they do function as members of the same group. I didn't know that some of them actually went to each other's weddings. They only tell me afterwards, or they send me a picture. So that's the society or the culture that I think we can create even for aspiring scientists. So, and one of the things we've done also in the past 10 years, Anjali was involved in this. She led this effort, you heard her name. So we decided to say, okay, scientists have to meet other scientists. And as aspiring scientists, our students need to meet other students so they can continue practicing how to talk to each other. How do they describe their data? If you get challenged at a local level, it's not too debilitating. You don't feel like, oh, I don't belong. And then if you get challenged at a regional meeting, you say, oh, I'm getting better at taking this, problem of being a challenge about my data. So we formed a group of us. The schools are listed there. Duke is involved, UMBC, VCU, Wake Forest, North Carolina, and Virginia Tech were the initial partners in this consortium that I cunningly called MAPPERS as the Mid-Atlantic Prep and IMSD Research Symposium. So the next meeting for our symposium is in Medical University of South Carolina in 2 weeks. So it has some legs, it's going, and it gives our students an opportunity to continue to expand their cultural interaction as scientists or aspiring scientists. So one thing that this concept that I'm advocating here is all about is that if the student is in your lab, give her or him the opportunity to experience something beyond the lab. I know I've had students, and I'll show you that in a minute. You're going to increase the likelihood that the student will succeed if you give that student the chance to be cultural outside of those immediate circle, the folks in your lab, your technician, your grad students, your undergraduate, because there are things that are important that we need to learn, right? So the student that we get, we sometimes don't know how they learn. We don't know if they give up, if they make mistakes. we don't know if they're afraid to, break an equipment or lose a really valuable sample because they are from a background that probably wouldn't afford that if they were going to be asked to repay. But nobody does that because science itself is around troubleshooting. So you have to make your own mistakes and then figure out what those mistakes are. So the culture needs to be explained to the student. Normally you don't do it in one sitting, right? You give the student a chance to maybe learn it if you guys are interacting casually. But I think interacting with more senior members of the academy or graduate students outside of their lab, they will learn these quickly. Don't be afraid to make mistakes. That's how you learn. That's how progress gets made. So, and I would advocate that for minorities, this is really key, right? There's always a question about imposter syndrome. I think beneath imposter syndrome is the fear to make mistakes that could be very costly. If I break this, what will happen? Or if I lose this sample, what will happen? I think that is something that's critical, especially in a genome lab. I spent several nights, when I was a postdoc pouring gels, and some of them would have bubbles, and then I'll throw it away. And then sometimes we'll break a glass. So Lamont never kicked me out. So I learned that you have to make those mistakes and you have to be in a lab that tolerates that. Well, we can make more labs do that if we are culturally aware of that fear that students have. So the opportunity then to go outside your lab and interact with people comes, of course, with some cost. And for us, the administrators of Virginia Tech have been really, really generous. They give us funding so we can fund what we call networking meals. So we go, this is our favorite place called the Network, the Virginia Tech Inn. This is a cohort from 4 years ago. She'll be here tomorrow to talk to, she's at Cornell now. We'll be here tomorrow to talk to our students. These are all from earlier cohorts that are now in their careers. She's assistant professor at Boston University, and she's a assistant dean here at Tech. And this was one of my colleagues. This is one of my colleagues that we started the program together way back in 2003, Dr. Jill Seibel. She's now a vice provost, and you've seen this picture from Mappers. So having these conversations in informal settings, I would argue, provides the trainees an opportunity to build confidence, self-identify as graduate students in challenging areas, and maybe check with each other how to handle the landlord, say, things that could be crippling in a small college town. Okay, so I've told you about the concept of culturally aware research education. I want to now focus on the differences. Why, when you're doing that, you have to focus on the differences that students bring to your lab. My favorite cartoon that comes from there's a textbook called The Heredity, and you guys probably know of it. So this is what we live on, right? So as a geneticist, your interest is in differences, right? And lately you've heard a lot of talk about mutants and the virus for COVID-19 is mutating, and a geneticist knows right there that okay, we're having a variation, right? We have differences. So this cartoon really captures this quite a bit. As an agricultural genome scientist, my interest in differences, in variation, is about which turkey produces more meat, for example. My PhD dissertation was about semen volume. So because turkeys now do not mate naturally. So Tom Savage had these lines where they were divergent, and you had those that produce more semen and those that produce less semen. So there's a difference right there. And my focus then, by the time I graduated, we had 4 papers trying to biochemically and statistically characterize those lines and come up with some discriminant functions. So that's what we do as geneticists, right? We should be at the forefront of arguing why differences should matter in training. So the data that we have, so I've been doing this for the last 20 years now at Tech, and before that, 8 years at Tuskegee. The data shows us that a student's motivation, resilience, commitment, citizenship, and teamwork are more reflective of their potential to succeed than the other measures that we use now. It's hard to measure some of this, right? So we can, I agree with the counter argument that these things are, but you can read between the lines in the recommendation letter. You can chat with the applicant. You can chat with the recommender, and these things will emerge. So in my own lab, differences have been really important, right? He's now a professor at Guangzhou University. He did some work with the chicken lines that I had inherited at one point, and then so Leo was able to work very well with these folks that with Kweku, primarily. Kweku was this guy was a prep scholar, and together they at least created an environment where others will appreciate male and female, and different races could work together. This is one of the graduations that time. Zhao Qing, I was holding her here when she got her PhD. Differences are not something we should fear, right? They're things we should embrace as geneticists and scientists, really, because people bring different opinions, and there's a lot of studies about that. One of the things we have done to manage the differences, and this is another concept I want to argue for here, to manage the differences, we use what is called a cohort approach. So we bring folks in together at the same time. So we bring them in, say in June, no classes, but we do a boot camp, and they are able to bond together. So each cohort will be a cohort of friends, right? If we take this cohort, she was in psychology, in biomedical engineering, in neuroengineering technically, immunology, and he was in biology, and this young man was in biomedical engineering. So they became really close friends, where they now, after Virginia Tech, they still attend each other's functions. She's at Cornell, he went to Syracuse, Minnesota, he stayed here to go to vet school. She's at Carnegie Mellon. And same thing with this group. This is one of our favorite groups here, right? So they're different. She's at UNC, Dominique. She came to us from Notre Dame, but she's at Michigan. And she came to us from Lafayette, and she's at Columbia in biomedical engineering. But they were probably one of the tightest cohorts we've seen. So they are different, but they supported each other about apartments, about things to do, places to go. And I really, really enjoyed how that concept that we've been working on for so long was working with them. She'll be here to speak. Hagar will be here to speak tomorrow. And she told me when Tasmin was defending her master's, they all showed up to listen to her. So clearly, one of the things they learned from Virginia Tech, I hope it was from us, was this approach that differences don't matter. Even if the discipline is not your discipline, if you support each other, things are going to happen that are good. And of course, what we've done is show cohorts that we are way back. Almost everybody in the top picture here now has a PhD. She's at NIH, and she's at NSF, so she's here. So a lot of these things really that I'm arguing for, it's because they've had results. We've had results where we've seen things happen. So I think focusing on the differences and then using the community outside of the lab could help with the success. So here's a slide that I want to use as a take-home message from this section of the talk. We have admitted people in the prep program with a GPA of 2.4 and up to 3.8, with a GRE that's considered between quantitative and verbal, that's 260, very low, right? So those folks have gone on to be very successful. I mean, I can tell you somebody with a 2.9 went to Yale and now he's doing superb. So even though the merit-based approach would have left these folks out, I think pay attention to the differences that I argue for. If you pay attention to those, I think you may find success. Things will happen. Okay. Another difference that I want to argue for scientists is that we allow our trainees to go to conferences that are different. As a grad student, and actually as a postdoc, I only went to discipline-specific conferences. They were actually fun. I mean, I drove from Corvallis to Colorado for a poultry science meeting, to Utah. It was in Logan, Utah, for a poultry science meeting. I was the most fun I've had for a long time. Well, it was the most fun I had at the time, as a grad student. We went to the rodeo in Colorado Springs. The grad student I drove with, his mom was in Colorado Springs. We spent the night there. But my argument here is to say, give the trainees a chance to go to non-discipline specific meetings, especially that are ethnically focused. And this conference here where we are at, we do recruiting, it's called ABRACMS, ABRCMS. So one thing it does is also capture our concept of community, right? If you expose them to differences, they learn how to do that. And people can ask them questions, and then it won't be too, they won't be afraid that somebody's just checking to see if they know something. So, and one of the things I've learned is under these informal interactions, you can train the student to quietly, without meaning to learn how to speak about their research. If we go to the cellar, it's a restaurant here, my favorite place to go to, and you study and you say, so what happened last week? What was your data like? And tell me a little bit. I don't know anything. So those interactions really enable the students to say, okay, even under challenging circumstances, I can talk the same way I'm talking to him now, or to these other people. So give them a chance to go to these meetings. And ABRACMS is very large, about 3,000. It also gives them a chance to see people who look like them, because it's a minority conference. There are presentations there, but it's non-discipline specific, just different disciplines all mixed up, multidisciplinary. Okay, so my goal then is to wrap this up. I have to talk about superheroes, and I've talked about cultural community building, differences that we need to pay attention to recruit students, and then I want to wrap up by talking about the superpowers that we have as scientists. So I told you, I got this concept from Zach. So as a scientist, when you get the PhD, according to Zach, you have superpowers. That's true, right? You can create, you can discover, you can write papers, you can inform people. When you have a faculty position, you even have additional superpowers. You have a lab, you can recruit people, you can help them design their research projects, and you can help them determine whether something is not good for them, whether this is not a career for them. But one of the things that you also want to do is you want to learn how to teach them how to get along with other people. So when I was in Alabama, what I did at Tuskegee was, compete for a grant with a bunch of other folks that were at Auburn University, University of Alabama, Birmingham, Research Genetics, this is here. And we put together a grant, and USDA funded it, to bring in high school students. And so for several years, we'll bring in these students, then they will spend 2 weeks learning about biotechnology, and then we'll bring them around, see labs, and some things happen. Some of that work we transferred to Virginia Tech, actually, when I moved in 2000. So we had a funding from Bernard Harris Foundation. So here's where my superpowers went, right? As a scientist, I was able to write a grant, and it got funded. And then for the region, we were able to bring in an astronaut to talk to these students. And the students, after they finished their work for the 2 weeks, they had new experiences. They had research presentation that they were guided by their leaders, who were also grad students and prep scholars. So clearly, having a superpower as a scientist, it's not just for your data or recruiting grad students. It could also be for shaping younger minds. If you start them early, like I showed you. And here we have a photo with the astronaut, Bernard Harris, when he visited Blacksburg. I think one of my highlights in Blacksburg. Okay, and so you are not sacrificing much, really, as a scientist. You could help build the confidence of these younger people, and you could help shape their paths. And so I just have a few here that I put in. Roseanne Brooks is now a program leader at the USDA. She sent me a wonderful email when she got her DVM PhD, because as a high school student, she led this effort where we, and the paper was eventually published. We tried out, at the time, genomics was just getting up. We tried getting DNA from buccal cells, so from the mouth, from animals. And so, especially pigs, at the time, people were worried, how can you, if you can stress baby pigs or even chickens. So Roseanne and her colleagues, Greg Touchting, they published this in Biotechnology Letters. It actually got, when I was preparing for this talk, I was surprised how many citations it had, because we are the first to do it, and it was done by the high school students. So that to me, and Roseanne has gone on now to become a scientist herself, and occasionally sends me little notes and say, about time you win this award. So that's something that I have really prided myself for, that we contributed to the journeys as here, and then somebody else published a paper in biotechnology, molecular and biotechnology education that's been cited in PLOS. So the undergraduate, we've done, the IMSD includes undergraduates, and similar arguments you can make. When Stephanie was here, Stephanie Hartman was an undergraduate in biochemistry and animal science. She wanted to go to vet school. 3 years in the lab, she published a paper, and she was on another one. She's the first author on this paper that was published. She decided to go to do a PhD at Duke. So Stephanie ended up doing a PhD at Duke, and now she's on a faculty in Wisconsin. So the argument then that I'm making is we can use our powers, our superpowers, to create new superheroes, and that is not something we should be afraid of. And an approach that we have used extensively for our programs is bringing back people like Stephanie, for example, who have done great things when they were here, and they are still doing great things. So our current trainees will see other superheroes, right? So the argument is You cannot see, you cannot be what you cannot see. And here one of our favorites, she was a program evaluator. We worked with her several years. We actually went to Puerto Rico when NIH budget could be used to go to program directors meeting in Puerto Rico. And so she got to meet evaluators and people doing similar work as a grad student. And after graduating from Virginia Tech, she went to the Navy. And we actually just put another proposal in to see if we can loop her back to Blacksburg to help us increase our evaluation capacity. So this is how this concept that Zach really made me think about, that I think we can use in our genomics education. So I want to wrap up. I was given 30 minutes. I want to wrap up by talking about my superheroes. If you read this article in Science that was done 5 years ago, Doing Science While Black, you know, I had some challenges in grad school. You know, after my master's in animal science, I decided to pursue a PhD in a virology lab. But you know, now I think back to it and I don't really blame the lab, the environment, because I thought, okay, he was probably pushing for tenure. So anytime I come from the dark room, they're checking my gels and picture, you know, and I said, I can't deal with this. So the man who saved me from that was Tom Savage. He said, okay, come back here, and fought for me. He doesn't know this, but I think I know he had to do a lot of fighting. Because I wasn't really, you couldn't consider me as a committed animal geneticist at the time. Even though at the end I published 4 papers before leaving, but Tom Savage really gave me the confidence to continue working and leave the noise behind. And also he really made me be aware that a scientist can be a normal human being, that you can have family. He will bring the dog to, Max will come with him to the turkey farm. So, and I've tried to do that. You saw pictures with folks in my house and we do things together. So when I graduated, I spent a year with Suleiman, and you read the piece that Caroline did. I actually learned so much from her. I mean, by the time I left, we published 1 paper, but just the work ethic and the fact that you can stand up to other people, right? So I do remember we were having a conversation one day. She said, oh, you're interrupting me. And I thought, oh, that's good. This is, you can't. You know, you have to wait on people. So, and my other superhero is Betty Graham. I mean, I tell you, my faculty career would probably not be shaped like this if it wasn't Betty Graham. I met her when we decided to try to put together at Tuskegee a conference on the Genome Project. And so she held my hand, and that with Betty Graham's help, we brought in National Academy members. The conference had over 500 people, and it was a big conference at Tuskegee, big arguments, David Botstein, Maynard Olson, Richard Myers. So I really credit her for shaping my path to this. After the conference, she helped me compete for a summer fellowship that would be split in 3 years, right? So every summer, so the NAGR gave me a grant where I will go to the University of Washington Genome Center to work for three months in the summer, and then the next year I go 3 months. So bring that back to Tuskegee so my students can, learn from it. And I also served on the genome study section, and I will always sort of sit by Betty Graham. See, if you're in the room, things happen, and Betty Graham will have little tips, right? And I put this here because I was really pleased to see that she's come a long way. She did some things as a Baylor graduate, and this link is from Baylor. So Betty said to me, say, you know what you need to do with your training grants? She said, create an ombudsman position. So this is Betty Graham, and this is, you know, way back. So I really admire her. She does fencing, so you have to be careful dealing with Betty. She's tough. Okay, my non-science superpowers, these are my parents. You know, one of the reasons differences matter to me. My dad was married to 6 wives, and there were 40 kids in the family. I learned a lot from Randy Grayson. If not for him, I would have gone elsewhere. I wouldn't, you know, when I was looking to leave Tuskegee. The family concept came from him, right? So if you're a family, you can fight, but you have to make up because you all have the same goal, you know, getting the family ahead. And this is another colleague of mine, Daniela, and she's accepted to join the group, and she's been really, really effective helping us continue the next iterations of these programs. And these are our students. You know, this guy just got promoted. As folks, postdoc, he came to us from Penn. We recruited him aggressively. West Virginian guy, Pittsburgh, and this is from Florida. So we have, and my non-science superpowers, this is my wife and my colleague, Luke, and a friend of ours at Tech, and my children. They've been patient. They've gone through a lot with their, you know, the loss of their mom in 2008, but they've done well. This young man and his wife are lawyers now. He's in law school. So this is my daughter. So she's the one who's keeping me really young. We go doing this scootering around town. And finally, this is, I get these from students, and it really makes me happy. I every now and then there's an email or a card or something, or a gift. Somebody left me a gift, and I go, okay. I'm not looking for more pay. You know, at 60, there are very few things you worry about. But for me, getting these thank you notes really makes me realize that these are, you know, these efforts are worth it, especially that they are doing so well. Okay, so to summarize, I think the foundation for culturally aware research education is, if you're only for yourself, what are you, right? So try to create a way where your trainee can go outside the lab. You know, don't just have the trainee stay in your lab. They can get the data. If they are feeling good, they will get the data. But encourage them to connect. Maybe go salsa dancing, you know, maybe go do something fun. And also, if somebody has a low GPA or low GRE score, yeah, it may not be that bad. Maybe take a phone and call somebody that they've worked with, even if it's not in science. And then really the superpower part, I think it's about inspiration. Find opportunities, send them to different conferences, bring alumni that look like them, or maybe act like them, or, you know, from the same region, because you're looking to inspire people so they can be really creative and be successful. Okay, these people provide funding for the things that we do. I do a lot of goofing around, eating with students, and thank you.
Thank you. We cannot give you applause because, you know, you can't be heard, but I think it was an interesting seminar. And I think one thing that's highlighted here is the importance of mentorship. You give credit to your mentors, and the students give credit to you as a mentor. And I think that's really what is important in my opinion. So the chat box is open for questions, and I'm actually not seeing any questions, although there are 3 messages. Here. So what main arguments would you give to faculty or resident to operationally change their graduate education training approach to more inclusiveness, to be more inclusive of these identities?
So the first part of the question was?
So actually, there's a whole series of questions which I kind of missed. So I should maybe start at the top and start with a question. Can you go to the chat box yourself?
Oh, okay.
The first is from Olga Ameral. I'm so glad I have the privilege of listening to this presentation. It's such an important point. Students have to be able to learn from their mistakes, and they have to be able to interact with others without fear. I try to do that with my students, but it's not easy when every micro-leader is accounted for. Communication builds curiosity, self-confidence, and the skill to troubleshoot and be resilient. Thanks for making this presentation available. I come from the human genetics area and studied in different countries. I was lucky. So it's not really a question, it's a statement. Another statement or question is that what I loved most about the IMSD prep was that it encouraged us to discover how cultural and individual identities can inspire research. What I find most troubling with current graduate education is that a lot of programs are trying to mold a scientific mindset that is either devoid or discarded with an individual's cultural identity, especially among programs that may not have had a lot of diversity and representation in their faculty ranks. What main arguments would you give to the faculty who are hesitant to operationally change their graduate education training programs to be more inclusive of these identities? So this is a question from Albert Hinman and is really focused on, inclusiveness and how to deal with this issue. And you should be able to give us some hints here.
Yeah. Well, we also have Maggie here. Maggie, you want to weigh in? We have people who also have experience with this. I tell you, so our own data, really, and we've tried to push people on this. Sometimes it works, sometimes it doesn't. Maybe give them a way they can take risks, right? So I think the university has to be a little bit supportive. So let's say the university has some soft funding that permits that faculty who is reluctant to take somebody with a 2.6 GPA. I mean, now we push people to do it because we have NIH money. We'll say, okay, you do 50% in the second year after they come in, they do well, take that chance. And that happens, right? So I think that the goal here is to say, be flexible with the criteria that you use to select or to admit somebody in your lab. Maggie, what do you think is that?
Well, I've sort of come to believe that the schools or departments, whatever your organizational unit is, probably needs a person who really can help the students get balanced, because I think it's so much to put on the back of an individual faculty member. I mean, for an individual faculty member, if you want to make your lab diverse, just smile more, just make people welcome when they show up. I mean, you can't believe what kids go through. But Ed and I have slightly different approaches, which I think are very complementary in terms of how we work with the students. I also think one of the things that's really, if you're going to talk about high-level changes, I think it's not helpful necessarily to only hear about the Meyerhoff program when you're talking about successes. Because if you look at our programs where Ed and I have both taken a lot of students that didn't have perfect GPAs and didn't have perfect backgrounds and have done very, very well, that only focusing on looking for high school kids that are high achieving and moving them forward, that's one way to go. But Albert Einstein and Alberta Einstein could be anywhere. So I think the faculty needs some help from the departments in terms of providing the support. So a faculty member could say something and a student could take it wrong. It's really great to be able to triangulate with a person who can say, he or she didn't really mean it that way. This is a way to look at it, and really help the student outside of the lab to develop the resilience so that they can move forward and really benefit from what the faculty member has to teach them in terms of science. That's
I tell you, there are many examples of how we've handled some of this. So you take the risk, and then something comes up. We do two things. We go to lunch with the student, and then we go to lunch with the professor. So the professor knows that we are all on the same team. So one of the concepts that I've developed, I didn't really argue about it too much here, is that team approach. So all this community is really based on team, right? So if the student has a team that includes me, that's outside of her department, she can come to me. Or if she finds me a little bit prickly because she wants to go to Germany for a meeting, I say you can't use NIH money, she can go to her advisor. So, and then when we had the ombudsman, we could use the ombudsman to handle it too, to help us get better. So that's another phrase that I throw around a lot. When these things happen, the opportunity is to get better, not to just say, hey, you're mean or yes. Does that help, Hugo?
Yeah. Doug, do you have a question? I see you've popped up there and you posed a question, so maybe you have a question.
Yeah, I do. I really enjoyed your talk. I have somewhere. The question is the following, which is, you can look at the students that are entering your program as being, undereducated about science and needing, some help in those categories. And I think that it extends more generally to the public. So I'm kind of wondering if there are lessons you can extrapolate. So for example, you started off with the idea that, science is about making mistakes. And I think a really good, where the public is missing this totally beyond your group, which is generally undereducated. Sorry about the thing in the background. Is, for example, we as scientists didn't get upset with the change in decision for Fauci for mask, because, it's a classic example. He had a certain amount of information about this virus, and, you put the mask on the people who are doing the service. Once they found out it was highly contagious and it had asymptomatic, that changed the whole thing. As a scientist, you say, okay, I have new facts. I made a mistake about masks. Now I'm going to move on forward. And so I think if the public understood better the concept of making mistakes as a part of the science, as what you're trying to teach your students, that would be really important. So I wonder if you talk to your students about what was the biggest thing that sort of helped them get over the concept of, making mistakes in science, and could we teach that to the general public? You could scale this. I think it's a bigger issue that is brought out by these particular students, but exists in science generally. So, I guess the question, have you talked to the students? Do you have a general idea about what's the biggest hurdles they have in terms of getting over the issue of scientists make mistakes? And, can we put that into some formal education or?
Yeah. So I think I briefly touched on it, and this is really where the cultural, the cultural aspect of science and the ethnicity comes in. I think the general public hears a lot about imposter syndrome, right? Which is I don't belong here. But really what in a science lab, what that is saying is, if I'm in your lab, I'm going to break things that are very expensive, and I don't want to be there and cause that, or I'll lose some samples. So what we have tried to do is to say, those things, everyone does that. If you don't make mistakes, then you are not doing it right. So you have to accept that something is wrong sometimes. And if they go right all the time, then this is not really science. You have to have some variation to create. But I tell you, this challenge for me, it's even in my home, right? I'm married to an English professor. So I have to sometimes convince her that, you know, that I have to change my mind about something, because the new evidence is, you know, the new thing that I've seen, you know, provides that confidence for me that I should change my mind. I've seen something different. Yeah. And so that's but that is the challenge that we have. I mean, I agree with you that the public, and I think the effort to get more minorities to be superheroes, you know, to have the superpowers that I talked about, will help. You know, will say, hey, somebody may die with, you know, from a vaccine. It may not be the vaccine, it may just be coincidence. So we'll be able to explain things. You know, I think the more of us that will do this, the wider the explanation for science will go out.
Maybe we can close with Maggie, who's been kind of eager to respond to Doug's question.
Yeah.
Well, I wanted to bring up another side of that, Doug, which was what you saw in the response to COVID was hierarchical bias in science. And there were people talking about masks very early, but they couldn't get their voices heard. And so I think that, you know, we have to look on both sides of the mirror here on this in terms of how do we improve what we do, what kinds of mistakes. And I think the thing is that I imagine for Ed and certainly for me, it's you know, we chose to be in places where we would run into the students that we really wanted to work with. And as a result, it was difficult sometimes to have voices heard. So how do we, since we know science that diversity is really important and biodiversity is important, how do we all take responsibility for really looking more deeply at how we can get more voices to be heard in terms of making the decisions. I think COVID's a great example of, you know, if you're not at Harvard, Yale, and Johns Hopkins, you're out. So it's, anyway.
Yeah, I just want to make clear that I probably didn't phrase myself very well, but what I was trying to say is that the lessons we learn from solving some of these diversity issues will have an impact on the general public way beyond the diversity issues. That's what I was trying to say. You know, the COVID issue that I was describing, the upsetness with Fauci wasn't you know, it's extended across all races, all cultures, because many people don't understand the concept that Ed was talking about, that science is, you know, is a process. And so, you know, the lessons we learn from diversity will, I think, have a good impact on the education of all Americans, or, you know.
So I'll just add, communication is always the hardest problem, it seems to me, and you're right. And so I hope that this helps, and Ed certainly has helped me a lot in thinking about this.
Well, on this positive note from Doug, Maggie, and Ed, I think we're nearing the end of the hour. And again, Ed, thank you very much for all you've done and for your mentorship. And we're happy you've been recognized by GSA for this. And I hope you've been happy to get this recognition.
I have been. Thank you. Thanks for hosting this chat.
Thank you.
Thanks, everyone. Appreciate it.